Good morning to everyone. Myself, Dr. T. Arun Kumar, Department of Chemistry, SNS College of Technology, Coimbatore. Today we are going to see about the topic structure, properties and examples of carbohydrates. So first we are going to see about the what is mean monosaccharide, structure, properties and examples. Monosaccharides are simple sugars. They have general formula as CH2ON. Monosaccharides are reducing sugar. The test for reducing sugar is called Benedict's test. They are sugars which taste sweet, are soluble in water and are insoluble in non-polar solvents. They exist in straight chains or in the ring or cycle forms. So blue color indicates that the sugar is none. The green or yellow precipitate indicates that traces of reducing sugar. Orange red precipitate indicates as a moderate and brick red precipitate large. They are classified according to the number of carbon atoms in the each molecules as triose, that is 3 carbon, tetrose, that is a 4 carbon, pentose, 5 carbon, hexose, 6 carbon, heptose, 7 carbon, and so on. The names of all sugars end with O's. The O's indicate that it is a sugar. Example, glyceraldehyde, we can call it a triose. That is, trio means 3, so it having 3 carbon. The next one, erythrose, that is a tetrose. Ose is a sugar and tetra means it is a 4 carbon. Then ribose, pentose, so pentose. So pent indicates it's a 5 carbon, then ose indicates a sugar, so 5 sugar. Then glucose, hexose, so hexose means hexa means 6, and then ose means sugar, it having a 6 carbon. Then fructose also comes under the hexose, that is 6 carbon. Galactose also comes under the hexose, 6 carbon. Pseudoheptulose, that is a heptose. The hepta means 7, ose means sugar. They are used as a source of energy in respiration. They are important building blocks for large molecules. Then disaccharide, the structure, properties and examples. Disaccharides are made up of 2 monosaccharide joined together by the condensation of reaction. So 2 monosaccharide joined together to form condensation. So what is my condensation means? Removal of water. When the 2 monosaccharide join together with the bond, it liberates the water. That bond is called as glycoacidic linkage. The condensation reaction is a joining of 2 molecules with the formation of new chemical bond, and a water molecule is released when the bond is formed. The glycosidic bond is formed between 2 monosaccharide if 1 carbon on one monosaccharide joined to form 4 on another monosaccharide. It is a 1,4 glycosidic bond. Nothing but in first monosaccharide, the first carbon joined with the next monosaccharide of 4th carbon. So that is called as a 1,4. Then bond is called as a glycosidic bond. Example, maltose. Maltose is formed from 2 glucose, that is alpha glucose. We already studied that we having alpha and beta. So in maltose we consider only alpha glucose. So alpha glucose molecules join together by the glycosidic bond. The sucrose is formed from the condensation reaction between the glucose molecule and the fructose molecule. The lactose is formed from glucose and galactose molecule. Sucrose is, we call it as a non-reducing sugar. The disaccharide can be split apart into 2 monosaccharide by breaking the glycosidic bond by adding water molecules. So when we joining, water is removing. When we need to split, we want to add water molecules, which is called as hydrolysis. Hydro means water, lysis means breakdown. So with the help of water, we are breakdowning the molecules. The water provided a hydroxyl OH and hydrogen H, which helps the glycosidic bond to break. The sucrose is a transport sugar and lactose is the sugar found in milk, which is an important constituent of the diet of young mammals. The sucrose, glucose plus fructose, lactose, galactose plus glucose, maltose, glucose plus glucose. The polysaccharide structure, properties and examples. The polysaccharides are polymers by combining many monosaccharide molecules. That is mean more than 2 monosaccharides are joined together to form the polysaccharide. This process also is called as a condensation reaction. The molecule with 3 to 10 sugar units are known as oligosaccharide, while molecules containing 11 or more monosaccharide are true polysaccharide. Polysaccharide do not taste sweet. When you consuming them polysaccharide, we feel that it is a tasteless because their molecules are so enormous. The majority of polysaccharide do not dissolve in water. So when we try to dissolve in water, it won't dissolve. Polysaccharide made slowly, and one kind of monosaccharide are called homopolysaccharide. The starch which those made more than one monosaccharide called heteropolysaccharide. The homopolysaccharide is called a starch, and heteropolysaccharide is a hyaluronic acid. That is a homopolysaccharide. Homo indicates the same, poly indicates the many, saccharide indicates the carbohydrates. So many same type of monosaccharide joined together to form the homopolysaccharide. In vice versa, in heteropolysaccharide, hetero means it's a different. So different type of monosaccharide are joined together to form the heteropolysaccharide. We want to remain that more than 2 is called as a polysaccharide. Example, source, cellulose we get it from the plant, starch that we call it amylose and amylopectin, glycogen. So source is plant, for amylose from plant, amylopectin from plant, but glycogen we can get it from the animal. Subunits we having the beta glucose. For amylose we having the alpha amylose, amylopectin, that is alpha amylose, glycogen, alpha amylose. Bonds 1,4, that is a, we told right, one monosaccharide joined with another monosaccharide, that is a linkage, that is a 1 to 4. Then amylose also 1 to 4, but amylopectin either it will join 1 to 4 or from 1 to 6. The glycogen same thing, 1 to 4 and 1 to 6. Branches, in cellulose we not observe any branches, but amylose also we won't observe any branches, but amylopectin every 20 subunits we can view the subunits. Same way glycogen every 10 subunits we can see one branch. So here it represents in the diagram, and shape also it will be shown in the diagram. Then starch is made up of long chains of alpha glucose, that is amylose and amylopectin. Glycogen is made up of alpha glucose linked together by glycosidic bonds. Cellulose is also made of many beta glucose molecules linked by glycosidic bonds between carbon 1 and 4. Starch is the main energy storage materials in plant. Glycogen is the main energy storage material in animals. Cellulose is the major component of the cell walls in plants. The test for starch is called as a iodine test. This is a reference. Thank you.